Hey everybody, in this video we're going to talk about protein digestion and absorption in a rather brief manner, but we'll still cover it pretty thoroughly, I think. If you guys enjoy the video, if you like it, if you truly do learn something from it, please consider liking, consider subscribing, and share the video to people who you think might enjoy as well. Okay, let's get started. So, protein digestion and absorption, it occurs within the actual stomach and the small intestine. Nothing occurs within the mouth, okay? It's only occurring at the level of the small intestine and the stomach. So if you guys remember from like my first video on the gastric system, within the actual stomach, right, peptides are going to stimulate the activity of these G cells. And these G cells will produce a hormone called gastrin. Gastrin will go and act on the chief cells and the parietal cells of the stomach. The parietal cells make hydrochloric acid, making the inside of the stomach very acidic, and the chief cells produce this protein digesting enzyme that is inactive called pepsinogen. The presence of hydrochloric acid will make the inside of this actual stomach super acidic, and that is going to denature pepsinogen in a way that it becomes active, okay? And this active pepsinogen will no longer be called pepsinogen, it'll be called pepsin. Now, notice that pepsin works best at around a pH of 1.8 to 3.5, but really we usually say that it's less than 3.5, okay? So here's going to be the overview of it. Here's going to be some enzymes, protein digestion enzymes in the actual stomach, digesting this peptide or this protein, turning it into smaller peptides, which will then go onto the small intestine. In the small intestine, primarily the duodenum and the jejunum and the ileum. Digestion occurs all throughout, but mainly the duodenum right now. You can see that the first thing that comes in is the peptides, and of course the chyme. The chyme, if you guys remember, is super acidic coming inside of this actual small intestine at first. Which means what? Which means it has a low pH. So this low pH and peptones, they're going to stimulate two cells, the I cells and the S cells of the small intestine. The I cells are going to secrete the hormone cholecystokinin, and the S cells are going to make secretin. Now, you guys know that the cholecystokinin will go ahead and cause gallbladder contraction, which will push the content of the gallbladder out. The other thing that the cholecystokinin will do is that it will cause the relaxation of this little sphincter here, the sphincter of Oddi, which means now all the contents that are inside of it can come out into the lumen of the small intestine. The other thing, and this is the main thing that cholecystokinin does, it goes to the pancreas, okay? And it'll act on the pancreas to secrete digestive enzymes. Those digestive enzymes will go through the pancreatic duct and they'll fuse with the actual bile duct, which will then fuse to the opening over here, the hepatopancreatic sphincter or opening, and then they'll come inside the small intestine lumen. Now, the other thing is that on top of the digestive enzymes, we have to sort of neutralize this actual pH. So what do? The secretin will act on the specific ductal cells on the pancreas to make bicarbonate and electrolytes and water, and it'll secrete those alongside the digestive enzymes into the small intestine lumen. So now, in response to the low pH and the peptones, we now have the bicarbonate and the digestive enzymes to kind of start functioning and start digesting. And you can see that again, we have this digestive enzyme that has been secreted now from the pancreas in an inactive form. It has to become activated. Once it's activated, it'll start digesting those little proteins we had into free amino acids and maybe some little peptides, and they will get absorbed. Okay? So that is the kind of story that we're going to be following along here. So let's first look at the stomach. At the level of the stomach, again, we had the hydrochloric acid being secreted. Hydrochloric acid is going to take this protein, denature it, okay? When you denature a protein, it just means that you're not actually destroying any bonds, you're just you're getting rid of the original folded state, okay? And of course, the pepsinogen is being secreted as well, and the pepsinogen, you can see, is being turned into pepsin with the help of this hydrochloric acid. But funnily enough, when pepsin becomes activated, it has the ability to activate the inactive pepsinogen itself. Either way, creating more pepsin. And this pepsin will start digesting this actual protein. Now, pepsin is an endopeptidase, meaning it will act within the actual protein, okay? It'll act in the amino acids that are in between. And it's actually going to cut most amino acids, like the dicarboxylic ones. So that'll be your aspartic acid and your glutamic acid. It'll cut out the aromatic amino acids. And it'll also cut some leucine and methionine, okay? But specifically, guys, it is acting on the C-terminus of all of these guys. So just so we're clear, let me give you an example. Let's say here's an amino acid, okay? Here's a protein right here. Here's a string of protein. And it's going to be made up of 3 amino acids: alanine, methionine, and glycine, okay? Now, if you guys remember, I said that this pepsin likes to cut methionine. So it's not going to cut the bond between alanine and methionine, and it's not going to cut the bond between methionine and glycine. It's not going to cut the bond between alanine and methionine, which means the only bond it's going to cut is going to be this one, okay? And specifically, it is doing it on the C-terminus end. What does that mean? You know how each amino acid has a C-terminus and an N-terminus? Well, this one, the glycine, its C-terminus is here, and its N-terminus is here. Methionine, its C-terminus is here, and its N-terminus is here, and the alanine C-terminus is here, and N-terminus is here. Now, when I say this guy, this pepsin, likes to digest the protein, specifically this methionine amino acid at the C-terminus, I mean that it's going to come at this part and cleave the bond there, okay? And it can do this for all of the other amino acids I listed here, like dicarboxylic, aromatic, leucine, and again, methionine. But the whole point is, okay, you don't have to know this, but the whole point is that you're going to create oligopeptides and large polypeptides and some free amino acids, okay, possibly, potentially. These guys will carry on towards the small intestine, specifically the first part, the duodenum, okay? Now, at the level of the duodenum, you guys remember that we were currently secreting what? We were secreting all the contents out of that hepatopancreatic sphincter, which included what? The digestive enzymes. What kind of digestive enzymes? Well, you know the pancreas, it creates a lot of proteases, and again, I talked about this in a previous video, but some of the proteases that it makes are going to be trypsinogen, chymotrypsinogen, procarboxypeptidases, A and B, and proelastase. But these guys, they are secreted in an inactive form, or else otherwise known as a zymogen form, okay? And so in order for them to be activated, they require specific enzymes. Trypsin, or I should say trypsinogen, will interact with this enzyme that is on the actual enterocytes, okay? It is actually secreted by the enterocytes in response to the cholecystokinin and secretin. It's called enteropeptidase, and the trypsinogen will be cleaved by the enteropeptidase to create an active trypsin. Trypsin, just like the pepsin we saw, has the ability to activate itself again, okay? So any inactive trypsinogens that will continue to come into the small intestine, the active trypsin will now be able to activate them, okay? What about the other ones? Well, the other ones will be acted on by this green enzyme. What's this green enzyme? It's trypsin, okay? Trypsin will cleave the chymotrypsinogen and turn it into chymotrypsin. Trypsin will also act on the procarboxypeptidase A and B and turn them into carboxypeptidase A and B. And of course, the proelastase, trypsin will turn it into elastase, okay? So that's pretty cool. Now, where do all of these guys act on these little proteins? Okay, well, great question. If you look at it, trypsin will act on the C-terminus of basic amino acids like lysine, like arginine, okay? And this will obviously end up creating smaller peptides and free amino acids. The chymotrypsin, it'll act on the C-terminus of aromatic amino acids. And it'll also act on histidine, methionine, and asparagine, okay? But again, the entire point is we're creating these smaller peptides and free amino acids. The procarboxypeptidases, they're a little funny. So after becoming activated and turning into carboxypeptidase A and B, they're zinc dependent, okay? Meaning that inside of their active site, they actually require zinc to function properly. And besides the point, the carboxypeptidase type A will cut amino acids on the C terminal for aromatic or allopathic neutral amino acids. Okay, what does that mean? That means that it basically is cutting any amino acid that is not basic or acidic. That's it. That's all it's doing, okay? So it'll cut any amino acid on the C-terminal side that is not basic or acidic, meaning it's neutral. The B isoform will cut the C-terminal of basic amino acids, okay? Only basic amino acids. And you guys remember that your basic amino acids are arginine, lysine, and histidine, okay? And this will result, the digestion with these guys will result in free amino acids as well. With the proelastase will, it is also an endopeptidase, just like pepsin, meaning that it will start cutting bonds, peptide bonds, adjacent to allopathic amino acids. And this can actually be found a lot in elastin. If you guys know, elastin is a protein, and well, elastase will be cutting a lot of this elastin protein that is made up of a lot of allopathic amino acids. If you guys remember, allopathic amino acids is just non-polar amino acids, okay? So, you know, things like glycine, like alanine, like methionine, like phenylalanine, all that stuff. Sorry, not phenylalanine. And again, digestion with those guys will give us some free amino acids as well, okay? Now, there is another thing I could add on here where at the level of the ileum, which is like almost the end of the small intestine, the brush border cells of the enterocytes themselves have these peptidases. So these ones are different from all the ones we've been talking about here. These guys were peptidases that were secreted by the pancreas. These guys are on the brush border membrane of the enterocytes themselves. And they can be dipeptidases, they can be aminopeptidases, and they can be tripeptidases. All that means is that, for example, the aminopeptidases, they cut from the amino side of the amino acid. They usually cut oligopeptides. The dipeptidases will cut a dipeptide into 2 free amino acids. The tripeptidases will cut a tripeptide into 1 free amino acid and a dipeptide, which could probably then be cleaved again by this dipeptidase. Okay. Either way, the whole goal here is to just create these amino acids and dipeptides and tripeptides so that we can go and digest them. Absorb them. How do we absorb them? Well, absorption usually occurs in the duodenum and the jejunum, or I should just say the proximal 1/3 of the small intestine. And it can occur differently for amino acids and peptides. Okay. So, for example, for peptides, they usually guys, they use a PepT1 system, okay, or the peptide transporter 1 system, where this peptide will come in only when this proton or this hydrogen ion comes in with it, okay? So this is a symporter mechanism. And when this peptide comes in, it'll get hydrolyzed inside of the enterocyte into amino acids, and then it'll make its way into the blood through specific transporters. What happens to the hydrogen? The hydrogen will then be pushed out through this special transporter, which will bring sodium in to correct the initial gradients of the ions. But then now we have an extra sodium in here. Well, not to worry, the sodium will get pushed out with the sodium-potassium transporter, restoring the ion gradients. Some of the peptides might go through a paracellular routes, meaning they will go through the tight junctions that are in between adjacent enterocytes, and they would make it into the bloodstream. And some amino acids might do that as well, but this occurs very, very, very low amounts. Another thing is that amino acids are absorbed differently than peptides. We already mentioned that. And I'm going to give you a very, 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 very simplified version of amino acid absorption because it is super complex and super long, and we'll probably have a dedicated video to it. So, with that said, majority of the amino acids are absorbed in an active transport mechanism, and they are sodium dependent, if you will. So here is a good example of it, where an amino acid will go into the cell only when a sodium ion has bound to this transporter, increasing its affinity for the amino acid, okay? And they both go inside of the cell together. And you guys know that when the amino acid comes in here, it will then travel from the blood, from the enterocyte into the actual blood. Okay, and then the sodium will then get pushed out with the sodium potassium ATPase. Awesome. Another way, and I think this is the final way that amino acids can be brought in, is by transcytosis, and special cells in the small intestine can do this, or have been proposed to do this. They're called M cells. They're basically taking in the amino acid by endocytosis, putting it in a vesicle, and then literally carrying it to the other side of this membrane, right? So this is the brush border membrane, this is the basolateral membrane. So they'll take it in from the brush border membrane, and then they'll release it out of the basolateral membrane to the blood. Okay, and that's like the major ways that amino acids and peptides are transported. Now, one thing I do want to mention is that because amino acids have different transporters, okay, but they still intermingle, meaning that different amino acids can use the exact same transporter, even though they have dedicated transporters sometimes. So this presents a problem. Like when you're ingesting an amino acid supplement, okay, you don't want to take too much of that because it can cause competition of one amino acid over the other and then impair the absorption of one rather than the other. Another thing that you should note is that peptides seem to be absorbed more readily than amino acids are, and that peptides in general, when they go into the enterocyte, they'll turn into amino acids. This pathway seems to be the major way that we are getting amino acids absorbed rather than this pathway. And this raises a question for whether or not people should be supplementing, for example, with branched chain amino acids rather than just having a regular protein shake, because the protein will then be broken down into peptides and then absorbed more readily than the amino acids, whereas the amino acids, not only will that cause competition with other amino acids, and they're just not absorbed as readily as peptides. So that's one thing to keep in mind. Other things to keep in mind for amino acid transport is that, for example, neutral amino acids are absorbed better than charged amino acids, so negative or positive, okay? And again, just to really stress the point, over 60% of protein absorption occurs as just small peptides rather than amino acids, okay? So you can see 60% of small peptides, which then get broken down to amino acids, and then the remaining 40% is just as free amino acids, okay? So that's really important and has implications for what you're going to supplement with for, optimal gains. But guys, after these amino acids are eventually inside of the blood, they will go ahead and they will circulate into the liver, and majority of them will be taken up into the liver except the branched chain amino acids. Those guys will be spared. They will make their way to the muscle to be used there. Now, that's where we're going to stop with the digestion and absorption of amino acids. Guys, we will have a dedicated video to what is going to happen to amino acids throughout the body and what does their metabolism look like. Perhaps even the metabolism of individual amino acids as well throughout the body. But that's going to finish it for this video. If you liked the video, please comment, please subscribe, and please like. And I'll see you in the next one.